Hello and hello to all the class 12 students. So let us now discuss about pre-fertilization events which occur inside the gynoecium of the flower, which is nothing but the female reproductive part of the flower. In our previous video, we have discussed about the pre-fertilization events which occur inside the androecium, and we have seen how pollen grains are formed within the anther of the flower. Okay, so in this video, we are going to talk about the structures and events which occur in the gynoecium of the flower. Okay, now if you look at the structure of a pistil of Hibiscus rosa sinensis flower, so Hibiscus rosa sinensis is nothing but the China rose over here. Right? If you look at the pistil of the China rose, you can clearly see that it has got three parts: stigma, style, and ovary. So stigma is the platform where the pollen grains land during pollination, and style is the long tubular structure which connects stigma to the ovary. So ovary is the bulbous structure at the bottom over here. Now remember, inside the ovary we have got ovules. Ovary contains ovules. Ovules are also called as megasporangia. Remember, inside the anther we had microsporangia. Inside the ovary we have megasporangia, also called as ovules. Now if you look at a particular ovule, right, our ovule or megasporangium, it is important for us to know the structure of a typical ovule. Okay, and inside the ovary we can have one ovule or many ovules. Okay, and inside the ovary, the ovules are attached to the ovarian wall with the help of placenta. Okay, now let us look at the structure of an ovule or megasporangium. So megasporangium is singular, megasporangia is plural. Okay, so if you look at the structure of an ovule, we can see many different parts of the ovule. So you should practice drawing this particular structure. And here you can see that there is a structure called funicle, which is connected to the placenta, which is present on the ovarian wall. And between funicle and the ovule, we have got hilum. Okay, so these are the two structures which connects the ovule to the ovary. Okay, and then you find that there are two integuments, outer integuments and inner integuments. These two integuments, they have got protective role. Right? These are protective envelopes. Integuments, they are there for protection. And who they protect is the embryo sac. Okay, so the embryo sac is the female gametophyte, which means it contains the female gamete. Right? Female gamete is present within the embryo sac. And embryo sac, it is protected by these two integuments. And between integuments and embryo sac, we have got a layer of cells, right? Many layers of cells called as nucellus. And nucellus cells, they have got reserve food materials. And these reserve food materials, they provide nutrition to the developing embryo sac. Now over here, you can see that integuments, it covers all the parts, all the sides of embryo sac except at the top over here, where the integuments, they do not meet. This end is called as the micropylar end, and the pole opposite to the micropylar end is called as the chalazal pole. Now this is very important. While drawing the structure of an ovule, you have to mention these two poles. Micropylar end is the region where the two integuments doesn't meet, and the pole opposite to micropylar end is called as chalazal pole. Okay, so I advise you to practice drawing this particular structure. Okay, next, let us talk about megasporogenesis. Right now, what is megasporogenesis? Over here, you can clearly see that I have written embryo sac, which is the female gametophyte. It contains the female gamete. It is formed from a particular cell called as a megaspore. Right? Embryo sac la karine chongyoresha, megaspore la cell chine chongyoredwa. So let us talk about that. Right? So megasporogenesis, it is the process of formation of megaspores from megaspore mother cell. MMC la karre megaspore mother cell la gorda. So the process of formation of megaspore, which will ultimately become the embryo sac, it is called as megasporogenesis, and it is formed from megaspore mother cell. Now the process is very simple. Right? At the micropylar end, at the region over here, the region over here, there are many cells of nucellus. One cell, it becomes megaspore mother cell, right? We have got nucellus over here, and over here we have got a large cell at the micropylar end, and it becomes the megaspore mother cell. I don't know how it becomes megaspore mother cell, but this cell, it is destined to become megaspore, right? Now this cell, it will specially divide through meiosis, and it will give rise to 4 daughter cells, right? And megaspore mother cell is diploid, whereas the 4 daughter cells, which are now called as megaspore tetrad, they are haploid. Is it clear? So megaspore mother cell, it is one of the cells of the nucellus which becomes megaspore mother cell, and megaspore mother cell, it divides through meiosis, giving rise to 4 megaspores or megaspore tetrad. Okay, and it happens at the micropylar end of the ovule. Ovule gi micropylar end la one of the nucellus cell kharcha dure la bimbana megaspore mother cell cha dure, and megaspore mother cell dikhachi it divides through meiosis, and khazu cell khazu chagre cell shicha and cell shija khare megaspore, right? And it is called as tetrad and shila ora, so megaspore tetrad cha dur. Now remember, only one out of these 4 megaspores, it will become functional, it will remain functional, whereas the 3, they will simply degenerate. So din alo lang cell shi chaswa megaspore mother cell ne megaspore shi raswa through meiosis, the shi nalo ne only one of the cell will remain functional, whereas the other 3 will simply degenerate. Okay, and this one functional megaspore will develop into female gametophyte or embryo sac. So this is gametophyte, sorry for the spelling mistake over here. So one of the megaspore which remains functional, it will develop into the female gametophyte or embryo sac. So here we can clearly see the process of megasporogenesis, the formation of megaspore from megaspore mother cell. And out of 4 megaspore over here, 1 megaspore will remain functional, and that megaspore will develop into embryo sac. Now let us look at the formation of embryo sac. So din alone cell chick di embryo sac chagrilaro di khandes chagu megi process di tayanda. Let us look at the formation of embryo sac. Now one of these cells which remains functional, inside that cell the nucleus divides. Only the nucleus divides, not the entire cell. Only the nucleus divides. It is called as free nuclear division, which means the nuclear division is not followed by cytokinesis or cell wall formation, right? So cell ginaulo nucleus chick divide chidir, nucleus chick ni chaswa. So there is formation of 2 nucleus inside one cell, and these two nucleus, they will move towards the opposite pole, and again there is another mitosis. It divides, right? And the nucleus divides, it gives rise to 4 nucleus, and then again it divides, it gives rise to 8 nucleuses or nucleus over here. After the formation of 8 nucleus within the cell, there is formation of, or there is a rearrangement of these nucleus, and there is formation of cell wall, cell membrane and cell wall. And the arrangement is such that the 3 nucleus over here at the micropylar end, they will become egg apparatus. Means 2 nucleus after the formation of cell wall, these 2 cells, they will form synergids, and 1 cell will become egg. And at the opposite pole, right, at the chalazal end, you have got 3 cells over here, and these 3 cells, they are called as antipodals. And 2 nucleus which remains in the middle, they become part of the central cell. Okay, so ultimately if you look at the structure of the embryo sac, so this is a mature embryo sac. This structure is very important again, right? One important structure was this one over here, the structure of an ovule. Another important structure is the structure of the embryo sac, right? So the embryo sac over here, you can clearly see that at the chalazal end, we have got antipodals. There are 3 cells over here, right? These are called as antipodals. And at the opposite end, at the micropylar end, you can see there are 3 cells over here. And these 3 cells, they are called as egg apparatus. I will show you later on. Okay, we have got the egg cell over here, and then we have got 2 synergids. And in the middle, we have got 1 large central cell, and this central cell contains 2 nucleus over here. Okay, so this is the mature embryo sac. Now, if you look at the structure of a mature embryo sac, it has got how many cells? There are 7 cells and 8 nucleus. So therefore, the mature embryo sac, it is said to said to be 7-celled, 8-nucleate. So now questions may come, right? It is commonly asked in board exams that why is the embryo sac or the mature embryo sac of flowering plants said to be 7-celled and 8-nucleate? Flowering plant now your embryo sac the 7-celled 8-nucleate kharjin lagors nan thiwa ligrta. You have to draw this particular diagram and then you have to show that it contains 7 cells. I will show you the 7 cells. There are 3 cells over here, antipodals, 3 antipodals, 3 cells in the egg apparatus, 2 synergids and 1 egg cell. It becomes 6. And then we have got the large central cell. So we have got total 7 cells. But how many nucleus are there? 1, 2, 3, 4, 5, 6, 7 and 8. So it has got 7 cells but total of 8 nucleus. So the mature embryo sac is said to be 7-celled and 8-nucleate. Is it clear? Now practice drawing this diagram as well. Now in this one, you have to see that the synergids, they have got cell wall thickenings over here. Cell membrane and cell wall thickenings over here, which gives rise to finger-like projections called as filiform apparatus. You can see hand-like structure over here. Right? And these are nothing but the cellular thickenings at the corner. And they are called as filiform apparatus. And the function of filiform apparatus is to guide the pollen tubes into the synergid during fertilization. Right? When pollen grain lands on the stigma, it germinates into pollen tube, and the pollen tube, it has to enter the embryo sac. And filiform apparatus is the structure which helps in guiding the pollen tube into the synergids. Is it clear? So that the fertilization can happen. So this is about the structure of mature embryo sac. So in the next video, we are going to discuss about pollination and what happens after pollination. Okay, thank you.